Hello students, we'll be studying biomolecules. Now before that you have to understand that in this biosphere there are a number of organisms present, plants, animals. From where do they derive the nutrition? From the soil. So, if elemental analysis per unit mass of any organism is done and compared with the Earth's crust, you will see the same elements are present in the soil as well as the organism. Now, let us do some comparative study of it. So, this chart shows the elements which are present in the Earth's crust and in the human. We are living organism. So, let's compare how much the elements present here. This is a percentage, in percentage, how much it's present in the Earth's crust. We are deriving the elements from the Earth's crust and making it molecule. So we have to study first what is the difference in the amount of the element in the Earth and in us or any living organism. There might be variation, very little variation. There will be little variation. Now hydrogen. Hydrogen in the Earth's crust is 0.1. In human it is 0.5. Carbon 0.03. In human 18.5. All the living organisms, they are made up of carbon. Carbon compounds are all biomolecules, not carbon dioxide, but biomolecules are all carbon compounds. See, so in human it is 18.5%. Oxygen 46.6, in human it is 65. Nitrogen very little in the Earth's crust. We are keeping all the nitrogen 3.3. Sulfur 0.03, human 0.3. Sodium 2.8 and human 0.2. Calcium 3.6, 1.5. Magnesium 2.1 and in human 0.1. Silicon is 27.7. In us there's no silicon, it is negligible. So this comparative chart, you must know how much is there in the Earth's crust and how much is present in us. Now, if you want to just find out the elements, calcium, magnesium, inorganic molecules, sulfate, phosphate, then what you have to do, take the living tissue, dehydrate, burn it, take the ash and analyze it to find the different elements. But you have to find the biomolecules. You have to carry out this experiment to find out the different biomolecules. Now I'll take a living tissue, maybe a liver, a piece of liver or a plant tissue. Take a small living tissue in the mortar, put it in mortar and put the piston. You have to break it. You have to make chutney out of it. Now add TC8, trichloroacetic acid. Formula for acetic acid, CS3COH. Remove the H and put chlorine. So Cl3CCOH, trichloroacetic acid, you put on the tissue and you press with the make chutney, like in the mortar and pestle. That is, you have to crush it completely, make a slurry of it. Now when it has been crushed properly, a slurry is made, you have to filter it. Because now all the molecules have been separated, you have to analyze one molecule after another. Many molecules find. So after making chutney of it, you have to filter it. Filter in a cheesecloth, a very fine cloth, you have to filter. Something will remain up and something will come down. Whatever is dissolved will come down, so that is acid soluble, it's a filtrate. Acid insoluble, that is retentate, it has been retained up. Now, biomolecules are of two types. The one which have formed polymer, like you take glucose, it gets simplified. It is just glucose, one molecule, but it will multiply to form glycogen in the liver. So your simple is glucose, complex is glycogen. So same way for other substances also. The one which are polymer, which are chain, which are many, will be written up. The one which is simplified, which has been dissolved in the acid, it will come down as filtrate. So if the size is big, if it is 10,000 Dalton, it is big, it cannot come down, it will remain up. If it is less, if it is 18 to 800 Daltons, it will come down, it will be there in the acid soluble, that is filtrate. Now, what will remain up? Proteins, they are polymer, they will remain up. Nucleic acid, polysaccharide, lipids will remain up. Now, lipid is not macromolecule. So, you just label this upper part more than 10,000 Dalton as macromolecule, bigger molecule. And the one which is less, which is 18 to 800, micromolecules, small ones, which have come down, they are small. Micro, macro. So macro are the big one which have formed polymer, which are chain. So protein is a polymer, it has formed chain. Nucleic acid, polysaccharide. Lipids is a micromolecule, but it will remain in the retentate. Why? Because you have broken the cells. When you have broken the cell membrane breaks. Now when the cell membrane breaks, it has tendency to curl and form vesicles, because you have studied lipids, it has got hydrophobic and hydrophilic structures which will form vesicles. So lipid, even though it is a micromolecule, it will not come down, it will curl to form behave like macro, but it is micro. So that lipid is the only micromolecule which is there in the retentate. It is micromolecule, but it has not come down. It should have come down in filtrate. No, it is micromolecule, still it has been in the retentate because it curls to form the vesicles. It is mostly present in the cell membrane. When broken, they curl to form vesicles. So this is, you have to remember. So now whatever is in the cell membrane is there in the retentate. Some bigger molecules from the cytoplasm also remains in the retentate, but the smaller particles in the cytoplasm will come down. So glucose, amino acid, nucleoside, nucleotide, they will come down. So let us analyze the macro and the micromolecule. I hope you have understood this experiment. It's very simple. Take a living tissue, that's a piece of liver, you crush it with mortar and pestle, put in trichloroacetic acid, make a slurry of it, filter it, something is retained, that is acid insoluble, and one which is acid soluble will become filtrate. You have to analyze. Here you get protein, nucleic acid, polysaccharide. Lipids are macromolecule, but they are micromolecule, but they will behave like macromolecule because they form a vesicle. Now the one which are in the retentate, their size is 10,000 Dalton. The one which is there in the filtrate, their size is 18 to 1800 Dalton. So you name it as micromolecule. The acid soluble has got mostly cytoplasmic composition, substance which are dissolved in the cytoplasm. So glucose, amino acid, nucleoside, nucleotide are present in the acid soluble substance. Amino acids. Now the macromolecules. Amino acid, the basic unit of protein. There are number of amino acids, but there are only 20 amino acids in us and therefore only 20 proteins. Let us see what amino acid is made up of. It is a substituted methane. Methane means CHHHH. Now it is substituted by hydrogen remains as it is. This part of all the amino acid is same. Hydrogen carboxylic group and amine group is same for all the amino acids. So here you have to remember alpha carbon. The carbon is alpha carbon, therefore known as alpha amino acid. So this carbon has got hydrogen carboxylic group and aminic group. Now here all the three are same. There is a R group for every protein of every amino acid. There's a R group which is different, and the properties is due to the difference in properties due to the difference in the R group. So let's see how R group can change. H, it can be H, it can be CH3, it can be CH2OH. It can be just hydrogen. So now this alpha carbon carboxylic group, hydrogen and amine group and hydrogen, this makes the protein glycine. It makes the protein glycine. Now there is a variation from hydrogen to methyl. So what it is formed? Alanine. Alanine is formed. Now here there is more variation. Hydroxyl methyl group is attached. Hydroxyl methyl group is attached. So what is formed? Serine. Serine. So various amino acids, alpha carbon is in the center and its 4 valencies are satisfied like this. So here 3 valencies are fixed. One is hydrogen carboxylic group and this is amine group. This is fixed. Only the variation in the R group. So R will vary. Whatever is attached to the R group, that protein will be formed. So here hydrogen, glycine, here alanine CH3, methyl group attached, alanine is formed, hydroxyl methyl is formed, serine is formed. So here these are the simple amino acids, alpha amino acid, because the carbon here is alpha. Now there are some amino acids which are acidic. They might be acidic, they might be basic, they might be neutral. So which is the acidic? Glutamic acid. Glutamic acid is acidic. The acid which is the amino acid which is acidic is glutamic acid. Glutamic acid is acidic. Acidic in nature. It is acidic in nature. So now what is basic in nature? Basic in nature. Lysine is basic in nature. Glutamic acid, acidic in nature. Lysine is basic in nature. Now some are neutral. That is, example is valine. Valine is neutral. So amino acid can be acidic, basic, neutral. Examples: glutamic acid, acidic. Lysine, it is basic. Valine, it is neutral. Now there are some amino acids which are aromatic in nature. Aromatic means it will have aroma. It will have aroma. So let's see which are having aroma. So tryptophan. Tryptophan has got aroma. Now phenylalanine. Phenylalanine has got aroma. Tryptophan has got aroma. Phenylalanine has got aroma. Tyrosine has got aroma. So these are the aromatic amino acids. What you have to remember is they are substituted methane. Means methane is what? CH, all the sides. Here the carbon is alpha carbon, therefore known as alpha amino acid. So this H is substituted by carboxylic group, COOH. COOH. This H remains as it is. Here this is substituted by NH2 amine group. Now this is R. This is R. There is variation only in this. Rest of the structure remains same. Whatever is R, the properties of amino acid is same because of these groups, and they are different because of this variable group. Now the amino acid has got amine and carboxylic group. Amine is basic and carboxylic is acidic in nature. So here the amino acid behaves differently in different pH. In acid it behaves like a base, in base it behaves like an acid. Look how. In acid the amine takes hydrogen from the acid and becomes NH3 and develops a positive charge and becomes ionizable. It becomes ion, so it is charged here, NH3 in the acid, because from acid it takes H and becomes NH3. Carboxylic group remains as it is, so here it becomes basic. In acid it behaves like a base. Now, in base, this amine NH2 remains as it is and the carboxylic group loses its H because base has got OH. So, this H is lost to it, forms H2O water. So, here now carboxylic group develops a negative charge. But when it is neutral, this carboxylic group gives its H to the amine groups. It develops the negative charge, amine develops the positive because it has formed NH3. So here it behaves as negative also, positive also. It is ionized from both the sides. So it has got both the charges because it has given H from here carboxylic to the amine group. So this has formed Zwitter ion. When both the charges are present in the same molecule, then it is known as Zwitter ion. So here amino acid has got the capability of forming Zwitter ion. Hermaphrodite. Both the charges are present in the same molecule. So that is Zwitter ion. Now, an amino acid has both a basic amine group and acidic carboxylic acid group. There is internal transfer of a hydrogen ion from the COH group 2, NH2 group to leave an ion with both a negative charge and positive charge. Now, lipids are fats and oil. So it depends upon the melting point whether it's fat or oil. Now, gingerly oil has got very low melting point. That is why you can use on your skin in severe winter also. So let's see what lipids are made up of. They are made up of carboxylic group, and that carboxylic group has R group, different R group. That R group can be methyl CH3, it can be methyl C2H5, or it can be CH2 many times, 1 to 19 times. So here you can see the simplest fatty acid, that is palmitic acid. Now this fatty acid, that is palmitic acid, has got the carboxylic group, that is COOH. Attached to it is methyl CH3, and CH2 it is 14 times. So here how many carbons are there? 14 carbon, 15, 16 carbons are present in palmitic acid. Now same way in arachidonic acid, 20 carbon are present including the carboxylic group. Now glycerol is another fatty acid. It has got 3 hydroxyl group. Therefore it is known as trihydroxypropane. 3 carbon, CH2OH, CHOH, CH2OH. So 3 carbon and 3 hydroxyl group. Therefore it is known as 3 trihydroxypropane, that is glycerol. Now, lipids can be formed by the combination of fatty acids and glycerol. When they combine, they form glycerides. When one glycerol, monoglyceride, 2 glycerol, triglyceride, 3 glycerol, triglycerides. Now, fatty acids can be saturated without double bond, or can be unsaturated with double or more carbon bond. Now you see the structure of triglyceride. It is combination, it's a fatty acid esterified by glycerol. Here in the center you can see the glycerol, 3 carbon, H2H. Now what happens? It was OH. H is removed, only O is remaining. H is removed, only O is remaining. H is removed, O is remaining. Now how the bonding took place? This is fatty acids, this is fatty acid, this is glycerol. So this is fatty acid. Fatty acid had carboxylic group COOH. So now CO is remaining. What happened to OH? H came out from here, OH came out from here. Water has been formed, H2 has been removed. So dehydration has taken place. H from glycerol and OH from the fatty acids has been removed to form water, and so the bonding could take place. Same way here also, H is removed, OH from the carboxylic group, water is removed. Same way here also, water is removed. This is how the bond has been formed. So this is known as fatty acid esterified by glycerol. So it is esterification which has taken place. So what has been formed? Triglycerides. So it has R1, R2, R3, that is fatty acids. Now some lipids, they have phosphorus and they have phosphorylated organic compound. So they form phospholipid. Phosphorus is present. So this is lecithin which is found in our cell membrane. So here you can see this is glycerol. Here this is fatty acids, fatty acid and glycerol. Now here phosphoric acid is attached by giving OH, and H from the glycerol water is removed and a bond is formed. Now this phosphoric acid attached to this phosphoric acid is the fatty acids, which has got the base nitrogen. So this lecithin is phospholipid, which is formed by phosphoric acid, nitrogenous base. You can see the nitrogenous base. You can see the phosphoric acid here, nitrogenous base here, alcohol and fatty acids here. 1, 2, 3, 4 fatty acids. So it is formed by this 4 structure. Now the tissues, the neural tissues, they have more complex fatty acids. Now cholesterol maintains the fluidity of the cell membrane. It's a type of lipid. Cholesterol is synthesized by liver or it is taken through food. Now the structure of cholesterol is such. The formula of cholesterol is C27H46O. So it is arranged in this way. Now you can see OH, there's only one oxygen. Then the sticks which are popping, that has got CH3, 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 CH3. So basically the structure looks like this. It's a kite shaped structure, big kite, a small kite, again a big kite, small kite and a broken kite. So you have to learn the structure of cholesterol this way. So here cholesterol, it is found in the cell membrane. It's a sterol, it's a steroid which is present in the cell membrane to give the fluidity in the cell membrane, and if it is in excess, it causes many problems. Now many living organisms, they have carbon compound which have heterocyclic rings. For example, the nitrogenous bases present in DNA and RNA, like adenine, guanine, cytosine, thymine, uracil. The nitrogenous bases, they have heterocyclic rings. When they are attached to the ribose sugar, then they become adenosine, they become nucleoside. When adenine attaches to the ribose sugar, it becomes adenosine, it is a nucleoside. And when it is attached to a phosphate group, when the phosphate group gets attaches to the sugar, esterifies the sugar, then it becomes adenylate acid, which is a nucleotide. So here the nitrogenous base gets attached to the sugar, ribose sugar to form nucleoside. It gets attached to the phosphate group to form nucleotide. Same way here the uracil is a nitrogenous base. When it gets attached to the ribose sugar, it becomes a nucleoside uridine. And when a phosphate group is attached, esterified to the sugar, then it forms a nucleotide. So here adenine becomes adenosine, it's a nucleoside. Guanine becomes guanosine, it's a nucleoside. Cytosine becomes cytidine, it's a nucleoside. Thymine becomes thymidine, it's a nucleoside. Uracil becomes uridine, it's a nucleoside. So all this nitrogenous bases, when it gets attached to the ribose sugar, it becomes a nucleoside. And when a phosphate group esterifies the sugar, then it becomes a nucleotide. So here adenine becomes adenosine, becomes adenylic acid, that is a nucleotide. Same way, guanine becomes guanylic acid. Cytosine, which has become cytidine, now becomes cytidilic acid. Thymidine becomes thymidilic acid. Uridine becomes uridilic acid. So they are all nucleotides. This is how a nitrogenous base gets attached to the ribose sugar and it forms a nucleoside, and then it gets esterified with the phosphate groups to form nucleotide. Now RNA and DNA, they are nucleotide, they are genetic material. Now, chemistry is a very interesting subject. You study different substance, you study different compounds, find out its structure. Now, in biochemistry, we study about related to biology. Now you know that the primary metabolites are very necessary for the living organisms. They are sugar, amino acids, fatty acids, and many more. They are primary metabolites. They are very necessary for our development, growth, reproduction. They are very necessary. Now, scientists have found out that there are some chemicals, there are some compounds which are found in plant cells, fungal cells, bacterial cells, which are not primary metabolites. So they have named it as secondary metabolites. Scientists have studied them and they have used for the human purpose, human medicines. Most of them have got medicinal values. They have got use for humans. So it makes it more important to study the secondary metabolites. So now this pigments are the coloring substance. When you eat bright yellow, red fruits, you get this carotenoids. They are precursor for A vitamin. They have health benefits. When you eat blueberries, blue and purple fruit, then you get anthocyanin. They also have health benefits. Now alkaloids, the example is morphine. They have use in medicine. They have effect on central nervous system. They are used during surgery, but otherwise also it has got medicinal value. It has got antifungal, antibacterial, antioxidant, anti-cancerous properties. Terpenoids, monoterpenes, diterpenes, they also have this antifungal, antibacterial, antioxidant, anti-cancerous properties. So it has got benefit for us. Now, essential oils, lemon oil, which is derived from the lemongrass, it helps to control cough and cold. It helps in digestive tract problems. It controls blood pressure. So all these properties are present in the lemongrass. We derive lemon oil from the lemongrass, and it has got the smell of the lemon, therefore named as lemongrass. Now toxins, these are toxins. Heparin, ricin, they are toxins, but they control the protein synthesis. They will not allow the cell to produce protein. So it is used to control the cancerous cells. So it is used in the as chemotherapy. They are used in controlling the cancerous cells. Lectins, concanavalin, drugs, vinblastine, curcumin, they all have anti-cancerous property. They control the growth of cancerous cells. And they all have other benefits also, antifungal, antibacterial, antioxidant property also. So all this forms the anti-cancerous drugs with other health benefits. Now here polymeric substance, polymers, Rubber, as you know, the scientists haven't really don't know how it benefits the plant, but it benefits us. It gives rubber, which has got many uses. Gum is also used by us, then cellulose is also used by us. So this polymeric substance produced by the plants, which are secondary metabolites, are used by us. Flavonoids, they also produced in plants. They have antifungal, antibacterial, antioxidant, anti-cancerous properties. Scents, perfumes are produced from the plants. Spices are edible and have health benefits. Antibiotics, very important for us, because whenever you have any infection, antibiotic controls that infection, and even after surgery, if antibiotics are taken, then the healing is quick. So all the secondary metabolites, which are not primary, but they help the plant for the ecological purposes. They help the plant to survive in the environment. They do not have an immunity system like humans, animals, so they develop these compounds which help them to survive in the ecology, in the environment, and we derive our health benefits from these compounds, which are secondary metabolites.